Hello everybody, I'm Kasper, I'm CEO of Proxi Biotech. First of all, thank you to the entire BII team for allowing us to pitch here today. So, at Proxi Biotech, we have developed a detoxification platform for detoxification of bacterial toxins. We call this the BID technology. We can take any bacterial toxin, detoxify it by use of our BID technology, and then we have a toxoid. This toxoid can be used as an antigen in a vaccine. And once you inject it with the vaccine, you are protected from the disease symptoms caused by the toxin. Now, toxoid-based vaccines is not a new invention. In fact, the idea of using toxoid-based vaccines has existed for almost 100 years. And the way we develop many of our vaccines today are still by the use of this ancient method, a method that's based on formaldehyde. And when you use formaldehyde, you create some major challenges, but some challenges that can't be avoided by the use of Proxi Biotech's BID technology. So, first of all, formaldehyde is a carcinogen. With the use of BID technology, we only use non-hazardous reagents. Second, formaldehyde detoxification is a reversible process, meaning that the toxoid can actually revert back to the toxic state. Using BID technology is completely permanent. It cannot revert back. And third, and most importantly, when you use formaldehyde, it results in a lot of modifications on the surface of the proteins, meaning that the immunogenicity you get is slow, it's not optimal. And due to this low immunogenicity, formaldehyde cannot be used to detoxify a range of bacterial toxins, including those causing whooping cough, botulism, and Clostridium difficile infection. Using BID technology, on the other hand, results in only very few modifications of the surface protein, leading to a high optimal immune response. So, to validate our technology, we have developed a vaccine candidate targeting Clostridium difficile infection, CDI. CDI is one of the most common hospital-associated infections and has a mortality rate above 8%. Furthermore, the economic burden is tremendous. Every year, we spend more than $10 billion just to treat the symptoms caused by CDI. And as you can see in the graph in the middle, this is not just a problem today. This is a problem that's going to increase in the future. Simultaneously, we also see a almost threefold increase in the market value, reaching more than $1.6 billion in 2026. Vaccines are going to account for a third of that market. So the vaccine candidate that Proxi Biotech has developed shows some very promising results. And to support my statement here, I brought you some data. Now, we're not digging into all the details here, but what it shows is that our toxoids get a very strong antibody recognition. We get a high immune response when tested in animal studies. They're very preserved, and they're safe. We see absolutely no toxicity left in the toxoids. And these are the four essential properties that a toxoid needs to consist or possess in order to be used as a vaccine antigen. We also know that there's a huge demand from the industry to get access to new technology like this. And we know it because 2 months ago, we went to the World Vaccine Congress in Washington, D.C., where we met with decision makers from some of the biggest vaccine companies in the world, including Pfizer, Merck, and Sanofi Pasteur. Now, Pfizer, they are leading with a phase 3 vaccine candidate targeting Clostridium difficile infection, CDI. And the technology they use today to produce their vaccines is very similar to the formaldehyde approach I just mentioned before. And that really complicates the manufacturing process. And they've expressed interest in implementing our BID technology in-house for production of their CID vaccines in the future. Merck, they have also expressed interest in implementing our BID technology in-house for the development of AMR-targeted vaccines. And finally, Sanofi Pasteur. They're also very interested in to get access to our technology for the production of various vaccines, in particular for the whooping cough vaccines. So, to validate our technology, we have designed an activity plan in alignment with the requests from the industry. In addition to our CDI vaccine candidate, we're going to apply our technology on the toxins causing diphtheria and whooping cough. And the reason for choosing these toxins is not to make a vaccine targeting the disease for the market. It rather serves two purposes. So first of all, it allows us to run quick and dirty studies in head-to-head with commercially available vaccines. And second, the toxins causing CDI, diphtheria, and whooping cough, they are all very different when looking at their composition and their size. If we can demonstrate applicability of our technology on all these toxins, meaning we can detoxify them and raise a high immune response in animal studies, we actually demonstrate that our technology can be applied to a very broad range of bacterial toxins. And reaching this point, our proof of concept inflection point in early 2020 allows us to engage with the industry. Following this, we are going to identify new vaccine candidates to develop. Our focus at the moment is on maturing the detoxification technology to detoxify bacterial toxins. However, there's absolutely no reason not to believe that this technology cannot be used in other areas, such as production of vaccines targeting viruses or venom. Hence, the technology has a very big potential. A big potential that's created by a very competent team. In addition to myself, we have René and we have Aria. René, he's a senior scientist at Statens Serum Institute, and he's also main inventor of the technology we use. He's an expert in bacterial toxin and has extensive experience in both supervision and planning of research activities. And then we have Aria. Aria is a PhD student at Statens Serum Institute and co-inventor of the technology. With a background in biotech engineering, he has a profound experience in the detoxification of bacterial toxins. So right now, can you switch me? Thank you. Okay, anyhow, we're looking for 3 million kroner that allows us to reach our proof of concept inflection point, bringing our technology to market. So if you find this interesting, and you think you're the person or you know the person who can help us to bring this to the next level, please contact us after the presentations or on our email address here. Thank you very much.
Thank you very much, Kasper. That sounds extremely promising for me.
It is very promising.
Now let's ask our experts what they think about that.
Thank you very much for a nice presentation, and we are of course also in dialogue.
Obviously.
So can you elaborate a little bit on your patent situation?
We can do that. So we own all the rights for the invention, and we are going to file next week for patent. Yeah. So we have the final draft now. We're just revising it final time, and then we're going to file it.
Okay. And why do you win the battle compared to your competitors?
We win the battle because the big pharma companies, who are the only ones who bring vaccines to the market, they have asked for this technology, basically. So we have talked with them, and they want this new technology for their production. So that's why we win.
I'm interested in the C. diff program and the discussion with Sanofi. I think Sanofi had a phase 2 failure with
phase 3 actually.
Phase 3 failure with their C. diff vaccine.
Yeah.
And the opportunity they want to pursue with you, I think you said, was whooping cough. I'm just wondering if they discussed, or you have an idea as to whether that program failed due to issues with the toxoid, and whether you could help with that.
Yeah, I know that. So we talked with multiple people from Pfizer and also from Sanofi. So Sanofi are not, what they said is they're not completely sure why they failed. But we can see when we align the technology that Pfizer used, Sanofi they use the formaldehyde approach completely. Pfizer they modified that a bit, and they get a better immune response. So that's one thing. And there's also something with the testing in the phase 3 clinical trials, with like the results they get there. Yeah.
Thank you, Kasper, for a very nice and clear presentation. It's a bit out of my comfort zone, but one question anyway. You mentioned that the toxoids, they attack the actual toxic compounds, so they don't attack the actual bacteria. Is that correct?
Yeah. It is specifically for the C. diff. So Clostridium difficile express 2 toxins, toxin A and toxin B, and they give rise to the disease symptoms. So what you want, we know, like patients who have a high immune response against these 2 toxins, they're protected from the symptoms, while patients who has like a very low level, they get the symptoms. Yeah.